My name is Sagi Snir. As this is part of the long program, so most of you have been in the retreat and know me as Sagi Quartet Snir, but this time I'll speaking actually on some project that not necessarily connected to quartet, although everything eventually boils down to quartets in phylogenetics. It's quite similar to the talk I gave last year on the same topic, but we have new results that I will get into. But since many of you have not been here last year, it's important. Anyway, it is dedicated to the memory of my beloved advisor Benny Shaw from Tel Aviv University that tragically passed away. Yeah, and he's actually even related somehow to this work. So talk outline. So we'll start with phylogenetics in general, and we'll introduce the JUMP model that is now a basis to quite an array of works that I'm pursuing. And by the JUMP model induces the genome as a continuous time marker process. And by that, we will reconstruct the prokaryotic phylogeny using the synteny index. That is some approach that we developed in the group and for quite a while, but it gives, but the new results just build on it all the time and add rigor into that approach. And I'll show results on real microbial data, prokaryotic data, and let's see if we will have some time for further results. Okay, so yeah, pursuit after analytical quantities, and these are the further results that I'm not. Let's hope we will get there. Okay, so you know this that was borrowed from from previous talk by Erin Molloy. She used quite the same slide. Was taken from her advisor Tandy Warner that was here. So this is a phylogeny, and it represents evolutionary history. Leaves represent living extant species. Internal nodes represent wait a second. Internal nodes represent extinct species. Branches represent evolutionary relationship. Now, in some classical evolutionary school results in a tree-like relationship that is, it's called vertical inheritance. However, within prokaryotes, in particular within prokaryotes that we focus today, some very significant biological processes result in a network relationship. And one such process is lateral gene transfer or horizontal gene transfer, which is the transfer of genetic material not through lineal descent, from parent to child. Okay, so lateral gene transfer is all the time found more and more to be so prominent and important in prokaryotic evolution. And as we know now that most of us is prokaryotes, okay, within our body with the microbiome and etc. So it shed some importance on all this process and the pursuit of some rigor in that process. So this is a very famous work by Welch et al. From 2002 that showed that between only 3 strains of E. coli, only 40% are shared, are common. This is a significant mechanism which bacteria acquire antibiotic resistance, toxicity, adaptation to niche, and whatever. However, it has become quite in agreement that even prokaryotic evolution, the main trend is vertical. Okay, adheres in large a tree shape, vertical inheritance that implies a tree shape history of evolution. Nevertheless, HGT, horizontal gene transfer, hampers phylogenetics. But first, can we use HGT for phylogenetics, or despite of the strong conflict? And why should we use HGT for phylogenetic, and why should we use HGT systematics or analytics at all? Let's try to answer that. So I will introduce the synteny index that we developed in our group back at 2014, and it goes something like that. So it compares the neighborhood of a specific gene in 2 genomes. We call that a COG. COG for people in genomics is very clear, but COG is the same or the autolog of a gene. Okay, so we see here the autolog G and G are autologs. They were inherited from the same common ancestor G somewhere up in the history. Okay, and we look at the neighborhoods of G at both genomes. And such a perception, such a view combines both gene content and order approaches for phylogenetic reconstruction. People have identified, people have noticed that genome dynamics, all these processes such as gene transfer, gene loss, gene duplication, this dynamics that is beyond point mutation that we all the time speak, these processes play a very important role in the evolution of the prokaryotes, and have used these the outcome, the footprints of gene content or gene order to build trees. However, all of them, the gene content and gene order, have their own disadvantages, and the synteny index that we have used for quite a while combines these two ideas and overcomes these shortcomings. And when you actually average this synteny index, not only over that gene G, rather over F and E and A and B, every such gene, you average that, it provides you some measure of distance and similarity between the genome. Okay? However, the question, so we use that, but the question was how we actually can model that rigorously. So we can say something, so we can we can not only to upgrade this approach into real systematics in the form of, if people heard here about model of evolution like the Jukes-Cantor and alike. So these are, or Kimura, these are strong theories of evolution. It's not only some intuition. So this is the average and can be used to construct trees. But as I said, we want some rigor. So Mike Steele is a very famous person in phylogenetics, in mathematical phylogenetics, and he came to visit my advisor Benny Shor in Tel Aviv. And I came to Mike and I said to him, hey Mike, consider that jump model. A genome G with large N genes. Not infinite, but large enough that we can ignore local, or we can, like is said here, converge in probability, and you will see that next. There are n+1 slots in the genome where a new gene can be inserted. n+1 slots, okay? And every gene, G, by a Poisson process, okay, with some constant rate, lambda jumps to another, to one of these n minus 1 slots. And the target position is chosen uniformly at random. Can we say something about that in order to arrive at some model, some rigor, some measure of similarity? Can we say something about the Syntyne index that we just introduced based on that model? So why the jump model? Yeah.
For your model, are you using signed orders or
No.
unsigned?
Unsigned.
You're not talking about which strand it's on.
No. It's everything very, very simple, as you can see. The jump model corresponds to several evolutionary processes such as translocation, gene gain, loss. And this is like Tandy's question was like invited. The simplicity of that modeling allows rigorous analysis that was not expected initially when we just started with that Syntyne index. The assumption of large genome allows asymptotic result, as will be shown. So we need that in order to ignore, like we said, locality, that we can ignore small effects. Okay, so if we consider a genome, and again, every gene is unique. There are no multiple copies of a gene. So it's very, very, at least here in this very basic modeling. So we can consider a genome as a permutation of genes. And a jump is some transformation from one permutation to another, okay? And that defines, the above modeling defines the genome as a continuous time Markov chain because we have not only, and the states of the model are all genome permutations, okay? And factorial genome permutation. And every such transition from one state to another is a jump. Okay, so we have very restricted transitions here in that model. But let's focus on the synteny index for a while. So a gene and the synteny index is defined by a neighborhood. So consider the following. So in the previous work, we modeled the gene neighborhood as a birth-death immigration process that I will very shortly skim now, where genes jump into or out of the neighborhood affects its composition. So, yeah.
Are you going to formally define the synteny index?
Yeah. Oh, didn't I? Oh, okay. So the synteny index. Sorry, because I tried to take out what I spoke last year and
Nobody was here last year. It's different people.
Yeah.
Completely different people.
Yeah, but I cannot give the same talk.
I know, but Sagi.
Yeah, Elias, I'm saying that. Okay.
Huh?
Okay. So so what is the synteny index? So we take, so every such gene, GI, that we considered in previous slide, has its neighbor. And we just consider how much that neighborhood or how many genes reside in that neighborhood. Okay, so take identical genomes. So every neighborhood defined by GI is identical. Okay, so let me do that quickly for those who, or maybe we go back.
It's just the size of the intersection of the two?
The size of the intersection, normalized. Okay, divide by 2k.
Is it the distance or similarity or
It's currently the similarity. Yeah. Thanks for all these questions. Let me go out. Let me go back. Yeah. Okay. So see here, G. You see little g. So and say k is 3. Say k is 3. So the 3 or the 2k, the sixth neighborhood of G is defined by the rectangle, that frame that you see. And now we take the guys that are common, that are shared between these two neighborhoods that is defined by G. Okay? And now you do that for every gene that is shared, and theoretically we say that since we have only jumps, so the genomes in terms of content are the same content, same genes. Okay? So you average the synteny index of G, you average that over the entire genome. Okay?
between 0 and 1, right? So
Yeah, it must be, yeah. It is all the time it is normalized. First by 2k, and then when you average, you of course you normalize. Yes, sure.
What happens when in one of the genomes you have events like huge duplications or repetitions or even transposons?
Okay.
Some models, right?
Yeah. So like I said, this is the theoretical model that in order to arrive at something rigorous, like in many cases in evolution, a model is very, very simple. And then once you get it, even that was for us initially like, for me it was like a magic that we did arrive at something rigorous, okay? And now you do apply, you will see in real data analysis, and I hope to be in time, you will see that it holds. And as always, it, for me, as I'm a model person, it always astonishes me that the simple model, not very far from realistic, or have strong, yeah, and yeah, it's this in particular, like, amazes me every time that it's so significant biologically. Okay, so please do stop me, and like I said, I took out, in order to be, to be fair, not to give the same. I would have spoken about quartets, but I want to I want to vary sometimes. Okay, so please do stop me if things are not clear. Okay, so this is the, we call it the genome permutation graph. But we return to focus on the neighborhood. And we focus at a specific neighborhood. Not only at a specific neighborhood, the neighborhood of G sub L, and some guy in the neighborhood G sub L prime. And remember that we have a rate lambda of jumps into every such slot, and a rate lambda of jump out of the neighborhood, outside the neighborhood. Okay? So, and we model that as a birthday immigration. Let's see how we actually model that. So when GH, G sub H, H for horizontal, jumps in, see what happened. It pushes G sub L prime out. Okay? Have a look at G sub L prime. It jumps out because GH was inserted by a jump. And when some other gene, GH, it's not the same, jumps out, then GL prime gets back, or gets into, back or not, whether it moves into the neighborhood. So this is, so we can model the distance. And if this is a death, and the previous insertion was a birth, we can model the distance between these guys, G sub L and G sub L prime, as a birth death, and we need an immigration here. We need an immigration here model. Okay, because a death is a jump out of every such guy, and a death, this is a death, and a birth is an insertion into every such slot. Okay, and let's denote x sub t, a random variable, x sub t, as the distance between GL and GL prime. And again, that distance is a birth, death, immigration model, or a death birth for you. Okay? So xt is a random variable that behaves as the size of the population. and it is affected, xt, the distance, is affected by the number of jumps into and out of the interval, okay, between GL and GL prime. And that applies to every GL prime in the neighborhood. So we need some average here over all the genes in the neighborhood. We need to average that. Now, since we have more jumps into, because we have more slots than, one more slot than genes inside the neighborhood, over time, distance between genes grows. So the neighborhood gets evacuated from, yeah. Okay, so now in that state model of the birth-death immigration, let Pij denote the probability of xt move from state j to be at state j, given that at time 0 it was at state I. Okay, what is the probability during time t to be at state j, given that we started at state I? All this is quite standard in birth-death theory or in Markov theory. And as I said, xt can be described as a birth-death immigration process. And the factor of that process is only lambda, the rate, and the time that is passed. So this is the model. This is the model. This is the single immigration, and this is death. These are death. These are death, jumps out. These are birth. These are birth. Okay. And this is how the neighborhood or the distance grows. This is how the distance grows.
Why do you take birth rates equal to death rates?
Because it's actually the same process. It is the jumps. So we have a rate lambda for jumps. So these are the same events, jumps, whether they are into or whether they are from your neighborhood. So it's the same events.
So the only reason you're using the birth death, it's kind of a different view on the state space, right? Because then you just see it like it's.
It's actually different states. Right. So the states, you have one model is this. Exactly. One model is this. This is this modeling the neighborhood, and the upper model is all the genome. Is the genome permutations. So these are two. This is more local. It is, and actually, it's not on a neighborhood, rather a distance between two specific guys in the neighborhood.
But if you want to look higher up, then all these processes are dependent between them. They are not, right? If you want to look at the overall thing as a collection of these processes, they are
yes, exactly. Exactly.
One more question. So what happens if one of the, here you're assuming that the two genes you're considering, none of them jumps out, right? That they remain static, but things can...
Yeah. In the analysis, later on, we will see that. Later on, it's part of the assumption that will be calculated. that assumption. I mean, that probability. We'll take into account in probability, yeah. Exactly what you ask. You know. So now, so this is the theorem, but again, it's not new. This is not new. It's from 2019. And the theorem is that the synteny index over the entire genes G0 and G sub t, where G sub t evolved from G0 over time t, is exactly this, Ilan, what was asked, that the two guys have not jumped. The two genes that we analyze have not jumped. And Q sub k, k size of the neighborhood, t. And that Q sub k, so this is the probability that both GL and GL prime have not jumped. Because if they jump, we lost all the information. And Q sub k of t is the probability by that local model that we defined that G sub L prime stays in the neighborhood, and it is averaged over all guys in the neighborhood. The guys that they were direct neighbors and father. So how many guys really stay in the neighborhood? And everything, and this is defined by Pij of t, that that transition probability in the neighborhood model, which is actually a function that is very tough function, but solely only on lambda and t. These are, and k, of course, k. Yeah. Yeah. So yeah, everything is yeah. All the, I mean, yeah, all these local assumptions are yeah, genes. I mean, in continuous time, nothing really happens together. Oh, you mean, yeah, of course. Yeah, genes are independent. Every simplifying assumption that could be is taken.
Yeah, but is it reasonable in reality?
Well, actually, very much amazing. Let's speak after, I mean, offline and on realistic, because yeah, like I said, I mean, I hope to have time to get to our. So we have some collaboration actually that is inflected or sparked from this. Yeah, about genomic islands, synteny blocks. Okay, so but this is tough. This is tough to arrive, and we could not calculate that. So this is easy, of course. Yeah, lambda and t, as I said. This was hard for us to really express, and that is why we came with some, as we could not solve the expression for Q sub k, and hence extract t. We want to extract t. Eventually, you want to extract t or lambda t that are confined from that expression. So we used heuristic, and we actually used properties for exponentially decaying system. It was quite neat heuristic, of course, analytic, but we could not really prove. It's not exactly based on the SI, and we came with that heuristic to extract lambda t, which is the distance, where DSI is what we see. I think it was 1 minus SI. Yeah, 1 minus SI was DSI. However, we were after the analytical expression, and actually you will see very soon that it's not only for the rigor, it actually proved much more realistic. Recall that the quantity we're after is lambda t, reflecting the expected number of event jumps occurred along the path. The path in the tree. The path separating in the tree, the two genomes are the leaves. So in the expression, in the analytical expression used to extract lambda t, we still have an unresolved component, q sub k. Using techniques, so we actually developed our own idea for that, but then we were notified that from the 50s, Carlin that worked on birth death theory, they had expressions for that rely on the theory of the spectral theory and orthogonal polynomials. We were not aware of that, but we could actually, from the beginning, we could use that to extract Pij t in that neighborhood, and hence since that. But at that time that we were notified, we had our own combinatorial proof for or expression to find that. And I want now to say something about additivity, and so additivity and consistency. So a measure, a distance measure, is called additive if we can translate its values along branches, okay, or between the differences or the footprints of that measure, such that it correlates to the number of events occurred. And I don't want to get much into detail, but you can, but there are, for instance, you have the Hamming distance, or when you run BLAST or all that stuff, you need to convert the Hamming distance, the differences, you need to convert it into some theory that will give you additivity, that will give you the events over every branch, that you can add the branches and get the distance. So in that expression that is translated from the SI, we need to show that SI is uniquely defined by every t. Because if we don't have that, if we can get from one SI several t's, we will not be able to say that the new measure that we have is actually a distance. So we need that one-to-one correspondence. And by showing that qk, since this is monotonically decreasing in t, as you can see, if this is monotonically decreasing, we are done. And this is what we showed. And this is a very, so additivity, consistency, all that stuff is very, very important. And if you don't have it, you run into very embarrassing conclusions in systematics. So this is something about rigor, about something that is some desirable property in systematics. And But the above discussion implies that SI can serve as a measure of distance between organisms based on genome dynamics. And genome dynamics, as I said, is everywhere, in your microbiome, in just everywhere. Such a distance has implications beyond tree construction. You want to be able to say that these guys, by their are such distant by their genome dynamics, and can be useful to trace rate, acceleration, deceleration. All that things are very important in real life as part of cultured bacteria, like I said, toxicity, antibiotics. All these things affect rate of evolution and stress, and you can see that by the genome dynamics of that organism that is under stress. Okay, so like I said, all this was very theoretical, but we need to know who is the autolog of that guy here in that genome. So some practical projects that we run in the lab is really build all these autologous or order autology databases that has also a lot of practical issues, but so that we need in order. But autologous databases are also, again, autologies is like everything in evolution. It's so important, and so this is one more aspect of that project. Let's see how I am. Yeah, I'm okay. But this is results on real microbial data, and I want to say something here. So we speak a lot about distance, and when we measure phylogenetic method, we speak about accuracy, and that accuracy is the distance between the trees. But sometimes these distances are very technical and mathematical, and they don't really express some intuitive biology. Sometimes quartet distance, it can be the Robinson fault, the symmetric edge distance, and several others. So we actually developed along the years something that is called the coloring or recoloring distance, and it goes something like that. So we have at NCBI what's called the NCBI taxonomy, and we use the NCBI taxonomy, the tree number 1, we use it as a reference, and we divide that tree, we divide it into 14 clades. A clade is a subtree. These clades are disjoint subtrees, okay? And we color every such clade. Color is something like, more conceptual, but we color every such clade by some unique color. So we have here 14 colors in the NC. So every clade, and these colors are we call them convex, okay? They don't intersect between each other by definition because they are clades. And now every leaf retains its original color, and we see how convex, how disjoint the colors in 3 other trees. So this is our analytical tree, the tree that we obtained with the new measure, okay? With the new measure, with the exactly how we transform the SI into lambda and t, okay? So that is the new measure. This is, as was denoted before, this is the 1 minus SI, because SI is similarity, so 1 minus SI is some hack, some heuristic to, and you see that all the distances are very, very, how would I say, like artificial. It's not realistic. It's not realistic. And this is the heuristic that we developed back then. And it has, this is log distances. So something here is really like erratic. And this is the realistic. And when my programmer showed me the length that he gets from the heuristic and the length that he gets from the new analytical model, I was really surprised to see that here the analytics, the simple model really reconstructs very realistic distances, as can be seen, can be compared to this 1 minus SI, 1 minus SI tree, and to the heuristic tree, that here we have some very extremely long branches. So this is one thing. Now, you may remember, or let's say that this way. So the NCBI tree, no one in bacteriology really takes this tree as a gold standard, and there is quite an acceptance, and you could see that also from Siava's talk last week. Yeah, sure. The tree now in acceptance is a tree by the prokaryotic. The bacterial tree is by a group from Australia. It's called the GTDB tree, but just a second, let's conclude that. So the 3 genome dynamic based. This is based on intuition and distances, maybe conserved genomes. I don't remember exactly how it's a taxonomy. It's not a tree. It's a taxonomy. And here we have all of them are based on the synteny index. So GD-based trees in general agree with the NCBI coloring. So there is a significant conservation of colors here. Nevertheless, some significant incongruencies found between the NCBI and the GD-based trees. I should correct here. The exact tree exhibits realistic, this is the exact, the analytical, the exact, not heuristic. Realistic branch lengths representing number of genome dynamic events expected, of course. So let me move, because if you want to send it to some biology journal, you cannot compare to the NCBI taxonomy. And people will tell you, hey, did you compare it to GTDB? So we took the same coloring from NCBI, the same coloring, and we painted the guys from the GTDB tree. This is the GTDB tree, a highly reliable bacterial tree based on core genes, not genome dynamics, core genes, 16S and alike. And it's not very clear from here because the coloring it's technical or visual things that we could not get that coloring, but there is a very highly agreement between these two trees and the NCBI. So this is about the synthetic index and the coloring and all that approach of the neighborhood. And I want to move now in 3 minutes to some follow-up project I've been pursuing with colleagues at the NIH, and it's called the neutral model of genomic islands. Now, what is a genomic island? A genomic island, or synteny blocks, are a cluster of adjacent genes, and it relates to a question was asked here about genes jumping together, normally acquired horizontally with distinct functions such as bacterial defense or pathogenicity. And a model for HGT, acquiring is HGT, allows us to study other genome dynamics, and such as this one, the synteny blocks or genomic islands. Now, by the jump model, we can assess the frequency of genomic islands of synteny blocks created by random or found by random. Let's see how this is. Example of such sequence conservation beyond operons include superoperons such as those encoding ribosomal proteins or CRISPR system, like clusters that travel together between organisms. But let's see what is the main idea. Any distance, again, what is distance? Distance is number of such events, such random events, such jumps. Any distance induces an island distribution. The frequency, how many islands we see of length 2. And what is island? Island is 2 guys, A and B, that occur AB here and AB in the other genome. So let's see how that distribution changes through the length. So if you have a probability 0.4 of every gene to jump, you will see most of the islands will be very, very short, okay? While here, if this is only 0.3, you will see more and more long, and everything here is natural. Everything is random. Nothing here is biology, okay? And if you have this length, okay, that probability of jump, you will see a much more flat distribution of islands. Many islands of length, not many, but some islands of length 80 and 60 that you will not see at all when you have that rate of jumps. And here I want to conclude. Genome dynamics events provide finer footprints than the conventional phylogenetic markers. It's just something to say that you have more signal in the genome dynamics in the shallow branches of the tree of life, and harnessing these dynamics is essential to leverage these footprints to systematics. This can be done by accurate statistical modeling of these processes and devise tractable approaches to trace them. So it's not only, we don't only need to model it accurately, we need to devise approaches to trace them, like finding this Q that we could not in the beginning, and in particular, tractable. So there are several steps here. Modeling, then finding the expression, and then calculating them. Results on real data should be confronted with other approaches and existing knowledge. Do come to me about practice how the model is really practical, because there was so much that we learned along this year and before about, like you see, the accurate distances. And I want to conclude. Where is Eliezer's name? Eliezer always should be here. Eliezer, you see your name here? Okay, always when I always in the acknowledgement, it's always Eliezer is there, but that is